During this time, I would like to request everyone to please check your audio and make sure everything is working okay. You can either connect to your computer audio, or if you wish to call in, the numbers are listed on the right side of the screen. Thank you for your patience. I'm going to play some music right now so that you can test for this audio. We will start our presentation in just a few minutes. Thank you so much everyone for your patience. I'm going to play back the music for just a little bit more for everyone to be able to do an audio check. Hello everyone, my name is Mansi and I would welcome you all to our webinar today. Firstly, I hope you and your loved ones are staying safe and healthy during these challenging times. We at Bio-Techne are monitoring the situation as it evolves globally. We are considered an essential business and are operational and open to helping our collaborators with their immediate service needs. Quickly diving in to the crux here, what is Pharma Assay Services? Pharma Assay Services is the only CRO specialized in visualizing and quantifying AAV and lentiviral-based gene and cell therapies in tissue microenvironment. This webinar will introduce our RNAscope and BaseScope in situ hybridization ISH services, our capabilities and workflow, especially the use of our quality control tissue bank for target validation. After that, we will dive into quantitative image analysis capabilities and a few case studies on how our services are contributing to and advancing therapeutic programs of hundreds of pharma and biotech companies worldwide. So getting straight to the question, what is RNAscope and how is it relevant to spatial transcriptomics? Traditional transcriptomic analyses have relied on bulk tissue analysis such as PCR, which provide an average level of gene expression in a sample but loses the spatial and single cell information. This is akin to a fruit smoothie, which can be a delicious drink, but you can only guess at what is in the smoothie. Advent of single cell transcriptomic analysis, such as single cell RNA-seq, have now been able to provide molecular information at the single cell level. And to come back to the fruit analogy, can now provide you the menu and tell you what fruit and how much of each fruit went into that smoothie. However, these techniques still don't provide you with how those cells are organized in the tissue context. And this is where spatial analysis comes into play. Single cell multiplexing spatial analysis that detect RNA can provide transcriptomic information with spatial organization while retaining single cell resolution. So now, instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. The RNAscope technology is an ideal spatial analysis tool to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single cell level. It consists of three parts: a unique target probe that ACD designs against your sequence of interest, signal amplification system that generates a high signal to noise ratio, and lastly, visualization of single RNA molecules as dots. The assay allows for spatial mapping of mRNA, long non-coding RNA, splice variants, and highly homologous sequences in cells and intact tissues, all of which can be visualized with either chromogenic or fluorescent labels. And the assay can be performed on a wide variety of sample types, including FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, and cultured cells. The growth and adoption of RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then, there have been over 2,600 peer-reviewed papers published using the RNAscope technology in numerous journals, including many top-tier ones. And the technology is highly relevant across multiple fields of research, with over 40% of publications in cancer research, followed by neuroscience and infectious diseases. Furthermore, the incorporation of RNAscope technology into single-cell RNA-seq workflows is demonstrated through the rapid growth of publications using the assay to confirm and spatially map single-cell RNA-seq data in the tissue context, with over 80% of single-cell papers in very high-impact journals, including Nature, Cell, and Science. Diving slightly deeper into how the technology works, the first key feature of the RNAscope technology is the probe design. We depict the oligonucleotide target-specific probes as Zs to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z complements and hybridizes to your target transcript. Top of the Z is the base for the amplification structure. When two Zs hybridize in tandem to the target sequence, it creates a unique binding site upon which a preamplifier can bind, and the amplification tree can be built. Standard RNAscope probe for target sequence of 1,000 bases or more will consist of 20 double Z pairs pulled together and designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. So here you will see two double Z probe pairs binding to your target sequence. As mentioned before, the bottom of the double Z pair binds to the target binding sequence, joined by a linker to the pre-amplifier binding sequence. So here we have the pre-amplifiers added as a part of the amplification step to the double Z probes, followed by amplifiers. And each amplifier can further bind multiple labeled probes sequentially, hybridizing to assemble a branching complex at each ZZ binding site. Labeled probes can contain a chromogenic enzyme, such as horseradish peroxidase or HRP, that generates a visible signal after chromogenic reaction, detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of signal under a fluorescent microscope. This signal amplification strategy allows for visualization of target RNAs as single dot, where each dot represents an individual RNA molecule. Just to talk a little bit about the specificity of the assay, should only one of the double Z probe pairs bind to the target sequence, we would not have the pre-amplifier bind to that Z that has been bound to the target sequence, and no amplification tree would form, hence hinting at the extreme sensitivity of the assay. Here we would like to show you the typical RNAscope experimental configuration. Provided that the target is expressed in the sample and protocol is followed as recommended, the RNAscope assay is guaranteed. Because tissue samples can be highly variable, proper sample preparation and the right controls are essential to an RNAscope experiment. Samples must be properly fixed and prepared so that there is good quality RNA in the sample and no background due to poor fixation. ACD provides guidelines on how tissues should be fixed for optimal performance of the assay. Here are serial sections from a human lung cancer sample on which we perform the manual red chromogenic assay. At the very left here, you see the negative control probe DAPB, which is a bacterial gene that is not expressed in intact tissue. As observed in this sample, there should be no signal from DAPB. Right here in the middle panel is a section from the same sample stained with our positive control probe for housekeeping gene PPIB or cyclophilin B. As observed with this sample, you want to see fairly uniform detection with positive control probe, indicating good quality RNA. Certain tissues may express the positive control probe at higher or lower levels, but in general, you want to see expression throughout the sample. With results from these two probes, we can have confidence in the test data we see in the right panel. As an example, I'm showing a section from the same lung cancer sample probed for immune checkpoint marker, PDL1 or programmed death ligand 1. PDL1 exhibits a wide range of expression in tumor tissues. In this human lung cancer sample, we observe strong punctate dots with the PDL1 probe, indicating expression of PDL1 in this tumor sample. Controls show us that there is good quality RNA throughout the tissue sample and that there is low to no background, so we can be confident that the localized pattern of expression we observed for PDL1 is in fact the true result. RNAscope technology includes two unique assays to detect RNA in the tissue context at single molecule sensitivity with single cell resolution. RNAscope, as seen here on the left, for mRNA or non-coding RNA greater than 300 nucleotides in length, and BaseScope, as seen here on the right, for exon junctions, splice variants, and highly homologous or short sequences. Both these assays are available in different flavors. RNAscope is available in automated and manual platforms in singleplex or duplex chromogenic formats, and up to 4-plex fluorescence format using automated and manual platforms. BaseScope assay is available on automated and manual platforms as singleplex chromogenic assay and as a manual assay for duplex detection. Next, moving on to the crux of this webinar, what is Pharma Assay Services and how our expert histopathology services can be of use for your research needs. As you will see, we can help with acquisition of tissue samples through our trusted chain of tissue providers, pre-qualification of these for RNAscope and BaseScope ISH assays. We also work with third-party CROs and can easily manage the embedding and sectioning of samples in your studies, stain for RNAscope or BaseScope assays, as well as provide quantitative assessment and provide a report of all the findings to you for sharing with your colleagues. Really quickly, just showing a little bit about our bandwidth here with the PAS group. We are an experienced lab staff of over 20 people, and we service 20 of the top 20 pharma companies. Our average slides delivered over a year are anywhere over 15,000, and we have customers available in greater than 20 countries. Our team includes both certified pathologists and image analysis specialists who can help with your fluorescent and chromogenic bright field acquisition of images. The PAS group helps enable drug discovery, starting at target discovery stage with target screening and expression analysis, all the way into phase 1 clinical trials and development of the drug. Just going a little bit deeper into this, how can we help with target validation? We can help with screening and validation of candidate targets based on disease and cell type specific expression. As you will see here in the images, the normal sample shown on the very left shows no expression for the target gene, whereas the image from the diseased patient shows very strong and robust staining with brown chromogen and in the stromal compartment only. As shown below here, we can quantify this and represent the data as percentage cells that show positivity in tumor versus stroma for the target, and also depict it as an H-score on a range of 0 to 400, providing a dynamic scale for seeing the expression in tumor versus stroma for your gene of interest. We can also be of help with biomarker assay development and have routinely done studies to evaluate candidate biomarker expression across diseased tissues and establish assay performance, as well as dynamic range using bioanalytical method validation guidelines. We can help with reproducibility, intermediate precision, specificity and sensitivity studies, as well as detecting the stability and robustness of the assay for this target. Here we would like to share with you how we can help with safety and toxicology tissue screening. You can leverage our pre-qualified normal tissues and tissue microarray for high sensitivity screening of your targets for preclinical ADME and safety assessment. PAS has also been routinely involved in early feasibility studies and assay development. We have a procedure that begins with assay prototype establishment, followed by precision and reproducibility testing. Once the images have been acquired, and based on discussions with our sponsors, we establish a pathologist guiding scoring criteria. All the findings in the method development are documented and shared as written SOP and study report, which includes optimized assay conditions. And in conjunction with our FAS team, the assay is then transferred to a recommended CLIA site, where we help their team establish and run the RNAscope assay on their instruments and follow it up with a cross-method validation to ensure the results across site have good concordance. As you can see here, in 2016, we collaborated with Leica Biosystems to develop such DX tests. And on the right here is shown the Advantage of RNAscope technology over other assays with respect to morphologic context, sensitivity, high specificity, and tissue requirements. Two examples of use of RNAscope assay used as a companion diagnostic in clinical trials have been shown over here. First one was where Merrimack used RNAscope for patient selection based on heregulin positivity. Our second collaboration was with Bayer in Germany, where RNAscope was utilized as a companion diagnostic and used for early screening for patient-specific tumor markers in different cancer types. And with that, I'm going to quickly jump into our capabilities and workflow here in the PAS group. As you can see on this slide, we have a variety of offerings, and projects can be designed a la carte to help with overflow needs at any step. That is staining only, analysis only, etc. We also have ancillary offerings which include H&E staining, FFPE sectioning, and helping to source tissues, which can be one of the most difficult aspects for researchers, as we have found through our collaborators. Just to give you a bird's eye view into our bandwidth here, we have 16 Leica and Ventana auto stainers, 3 Aperio 82 bright field scanners, a Zeiss fluorescent scanner, and 2 3D Histech panoramic scanners across our headquarters sites. We also have 6 Halo workstations, which enable high throughput of 10,000 plus slides delivered every year. This is just to show you the bandwidth and how much of bandwidth we have here to take on your research projects and help you with those. Just to give you a little bit of an idea as to how you can leverage our pre-qualified tissue bank, we have depicted over here the different kind of RNAscope and BaseScope pre-validated tissues that are available through our bank. These ensure high-quality samples are provided to you with faster turnaround and guaranteed RNA integrity. ACD can also help source these tissues without a need for you to set up third-party vendor contracts and get into the purchasing demands of these. For new sample collection, ACD will coordinate with embedding CRO to ensure samples are prepared according to guidelines for best RNA preservation. The take-home message here being we can help you eliminate risk with RNA quality in the tissue, shorten the turnaround time for data to be getting into your hands, and reduce costs. Another offering that we have here is QC services. What this means is we can do quality check on tissues for the purpose of your research needs. This can help maximize the number of samples that pass QC by entrusting the RNAscope experts here on our side and help free up your instruments for other projects. What you would receive is QC'd samples with optimized assay conditions and H&E staining. Lastly, I would also like to touch upon the quality systems and security measures that we use here on our site. Our practices are based on GCLP-based guidelines, that is good clinical lab practice guidelines. We have defined sample management workflows and SOPs that govern the PAS workflow, including staining procedures, sample tracking, data management, and data QC. HeLa cell technical control is run with positive and negative controls in every and each assay run with strict run suitability requirements. Should any run show that the HeLa cell pellets fail QC, we would repeat those experiments. We have quality control workflows set up and ensure data review so that you get the best data in your hands. We also use internal reference controls based on sponsor needs and as indicated in our statements of work. Our regulated products are ISO 13485 certified and monitored under our quality management system. With respect to security, our files are shared using a secure enterprise file sharing system called Ignite. We have restricted data access and all the data is managed electronically. Just to show you in brief what our PAS project workflow looks like, the first step in the project is called phase 1 or sample QC phase. This is to test the tissues for RNA integrity, and our scientists perform multiple rounds of pretreatment to ensure the best signal to noise ratio. Internal controls provide confidence that maximum sensitivity is achieved when we run the marker staining as a part of phase 2. We also provide whole slide scanning for both the slides run in phase 1 and phase 2, and these are available for review at your leisure. Lastly, depending on your scoring needs, we can perform the analysis through visual scoring or data analytics methods using automated software. We would report this for you and via teleconference, share the data and have a discussion with our scientific team. Quality processes and SOPs ensure reliability and confidence in the data. On the note of data, here we would like to share with you some of the different scoring strategies that we have built for RNAscope and BaseScope assays. First scoring methodology that we utilize here is called a semi-quantitative scoring. This is performed on a 0 to 4 scales by our highly trained experts. This is a rapid scoring methodology that shortens the turnaround time and speeds the delivery of results. It provides expression level at low, medium, high, and also percentage of cells across a broad range. Next, we can also help with digital image analysis needs. It provides quantitative cell-by-cell gene expression data. Our experts here can help identify tissue types or regions of interest for your research needs and use built-in classifiers for annotation. Additional heat maps can also be provided for full tissue spatial expression. The digital data analysis platform that we use here is called HALO, which is provided by Indica Labs. HALO, while automated, needs expert scientist input for nuclear outline detection, followed by optimal cell segmentation, clubbed with appropriate ISH dot identification to ensure that the right dots are being identified in the right cells and provide you with true and accurate data. HALO provides interactive real-time readouts of individual cell data. The SOPs that we have built ensure that data are quality controlled at multiple steps, confirming that the scores reflect the staining patterns observed and that tissue artifacts are excluded. The data is reported as cell-by-cell results with average number of dots per cell, percentage of cells with positive signal, and total number of cells per region of interest. And finally, as an H-score. With years of experience and having developed and built, beta tested some of the modules for ISH identification, our image analysis experts already have pre-identified settings to give them a head start and provide this data to you in a timely fashion. We also have research pathologists and board certified pathologists on our team who can help with image annotations to suit your needs. For example, in tumor tissues and for identification of heterogeneity in expression patterns, our pathologist can identify tumor compartments, tumor and tumor-associated stroma, and just tumor-associated stroma alone to help evaluate the gene expression patterns in these different regions individually. Histopathology notes also include tumor load and visual aid scoring. Our pathologists are available for teleconferences and for data representation with review. Lastly, just touching on what our deliverables include. The stain slides that are a part of the study and any remaining unused material are provided back to you. We also provide a summary of the scoring results and, if appropriate, tissue or TMA heat maps. All of this is reported as a part of a study report. This includes representative images of ISH staining and HALO image masks. We also share with you the whole slide digital scans acquired at 40x, along with the imaging software needed to view these. All of this data can be easily presented by our scientific team via teleconference to you and discuss the next steps. Finally, coming on to some of the case studies and how we have been able to help our pharma clients progress their needs. The first one here is a case study in biomarker evaluation of IDO1. This was presented by Insight at 2018 ASCO SITC. IDO1 is essential to primary and rate-limiting step in tryptophan catabolism, required to generate chyme. Decrease of local tryptophan and increase of chyme can result in suppression of anti-tumor immune responses. Stellar cells, as you can see, were stimulated here with interferon gamma at varying concentrations for 18 hours and assayed for IDO1 expression. This was done using RNAscope ISH or IHC and also using Western blot. As you can see, in the top panel and middle panel here, the IDO1 mRNA by RNAscope ISH showed robust expression from 0 picogram per mil induction all the way up to 50,000 picogram per mil. Whereas when the same experiment was repeated using antibody for IHC purposes, not so robust staining was seen. Hence, it was readily observed that the antibody for IDO1 was not as sensitive as RNA-ish. Quantitative comparison of the three platforms, that is RNAscope, IHC, and Western blot, demonstrated that IHC was 10x less sensitive than RNAscope and Western blot analyses. A strong positive correlation between RNAscope and ISH Western blot was observed as seen in the graph here on the bottom, suggesting a highly coordinated induction of IDO1 by interferon gamma at both the mRNA and protein levels. Here we would like to show you the comparison of IDO1 IHC in comparison with IDO1 ISH using anti-IDO1 antibody clone SP260 or RNAscope ISH in melanoma. You can see it is quite obvious that the image on the left-hand side, where red dots can be seen representing IDO1 using RNAscope assay, are much more prevalent as compared to the staining performed using the IDO1 antibody. A scoring methodology was developed to classify IDO1 expression in tumor cells, immune cells, and the immune cells infiltrating the tumor in the peritumoral region. This was done in concordance with the pathologist at Insight and our pathologist here. An 8 score of 5 or greater was used as an arbitrary cutoff for IDO1 positivity. As seen previously, this quantitative approach confirmed that the sensitivity with RNAscope was much higher as compared to IHC. While only 24% of the cells showed positivity in the tumor region using anti-IDO1 antibody, RNAscope ISH showed 56% positivity in the tumor region. Similarly, an 8% higher positivity was seen with IDO1 in intratumoral region as well. Since the study aimed at identification of tumor types that express IDO1 and may be susceptible to treatment with IDO1 inhibitors, the use of RNAscope technology showed that a higher population of patients would be considered responsive. Moving into our second case study here, which was to study the immune oncological checkpoint target profiling in non-small cell lung tissue. To interrogate the co-expression patterns of various checkpoint targets in the tumor microenvironment, we first generated TMAs from lung cancer patients. Multiple FFPE tumor and normal lung tissue blocks were obtained and tested for RNA quality using RNAscope PPIB positive control and bacterial DAPP negative control probes. Of the RNAscope validated tissue, 60 tissues were selected, which included 54 non-small cell lung tissues and 4 adjacent normal tissues. These were aligned as 2 TME slides, as seen in the schematic illustration over here, where the yellow circles represent the tumor tissues and the white circles represent the normal tissues. We first evaluated PD-L1 gene expression profile by singleplex RNAscope, followed by evaluating co-expression profiles of PD-L1 with either other checkpoint markers or immune cell and functional markers in the tumor microenvironment by RNAscope Duplex and multiplex assays. What is demonstrated over here is the dynamic expression range for PD-L1. As you can see on the top left corner, core 2b2 clearly demonstrates high PD-L1 expression seen as red dots. And at the bottom right corner, core 2c4 shows absolutely no or very low PD-L1 expression. In between cores 1G3 and 2G3, we see medium high and medium low PD-L1 expression. Another finding we observed, besides the range of expression across the various cores, was a range of expression within each core. In this image, the tumor displayed differing levels of PD-L1, visualized as green dots, ranging from low to medium and high, pointing to an intra-tumor heterogeneous expression pattern for PD-L1. While this was all observed visually, we also wanted to share how RNAscope can be used to tease out quantitative nature of expression patterns with respect to heterogeneity in gene expression within the tumor. We performed duplex chromogenic stain for PD-L1 and LAG3, two very common targets in immuno-oncology, and found that in this tumor, PD-L1 is primarily expressed in the tumor compartment, whereas LAG3 is primarily expressed in stromal compartments. Halo algorithms classifiers were used for establishing 3 layers for analysis. Layer 1 happened to be the stromal region, layer 2 was established as a tumor region, and a third layer where the whole core was assessed. We found that with the region-based analysis, PD-L1 primarily was present in the tumor region at 62% positivity, and LAG3 was primarily present in the stromal region with 44% positivity. This information was lost, as you can see with the red bars on the rightmost corner, in a whole core analysis, where not very high differences were observed between the two genes LAG3 and PD-L1. An example of halo image analysis performed on immune checkpoint marker is shown here, where TIM3 was studied in non-small cell lung cancer tissues. As you can see here, RNAscope can be used to distinguish cold versus hot tumors based on low, medium, or high staining of immune checkpoint marker, TIM3 in this case. The heat map at the top shows the variability and range of expression of TIM3 across the different cores that were studied in this TMA. Lastly, with respect to this study, we were able to assess the co-expression of immune checkpoint markers at single cell level, which could potentially aid in understanding resistance mechanisms of checkpoint inhibitors, as well as bring insight into combinatorial therapies. This example illustrates the single cell co-expression of TIM3 and PD-L1 in red and green respectively, as indicated by the arrowheads. I wanted to really quickly touch upon dual RNAscope ISH IHC or IF assessment. That is a great tool that can help characterize cellular gene expression and is used as a combination of ISH with IHC on the same slide to simultaneously detect RNA and protein. What we refer to as dual ISH IHC can be used to characterize cell type specific expression, identify origin of secreted proteins, visualize cell surface markers with RNA of interest. It can also help visualize RNA binding proteins and their target RNA and dissect the regulation of gene expression. Because of similar workflows between ISH and IHC, including sample fixation, pretreatment, probe hybridization, signal detection, and data analysis, as well as the unique benefits of each assay described earlier, ISH and IHC are ideal to combine into one workflow in which the RNAscope ISH assay is performed first and then followed up by the IHC assay. We have expert recommendations from our scientists on site to ensure success with your dual ISH IHC assay. Firstly, it is recommended to combine working RNAscope protocol with a working IHC protocol. In addition, working with an antibody and a protocol that is already known and already established helps fast track this process. It is advisable to perform ISH first, followed by IHC. And third, it is recommended to optimize the IHC assay separately using RNAscope pretreatment reagents to ensure your protein will still be detected following RNAscope pretreatment. We have also found that the dual ISH IHC assay works better for highly expressed proteins due to the protease pretreatment that is used during the ISH protocol. Since you would be sending your studies here, quite a few of these recommendations are taken care of by our scientists here at the back end. Jumping into one of the examples of CAR-T infiltration that can be studied using ISH IHC platform. What is shown over here is that CAR-T cells or TCR positive cells, TCR T cells can be visualized using RNAscope assays. Since both of these are comprised of portions of various human proteins, there is almost no way to distinguish these from endogenous proteins using antibodies. RNAscope technology helps enable the visualization of CAR-T cells and TCR cell distribution in tumor microenvironment. While co-staining with a marker of activation such as granzyme B or interferon gamma helps enable the assessment of activation state, co-staining with a cell type marker such as CD8, CD4, CD3, etc. can help enable assessment of functionality and helps monitor anti-tumor activity. Here is a study that was presented by Bellicum, our collaborators at ASCO GI in 2020, where different metastases from 3 patients to lymph node, liver, and omentum were studied, and assays were performed on baseline and on-treatment samples. It was shown that lymphocytes in the tumor stayed relatively same between the baseline and on-treatment samples, whereas there was infiltration of CAR-positive cells that was observed in the tumor tissues in all on-treatment samples. It is depicted here in the rightmost image as red dots, which show CAR positivity in the sample, and green dots, which show CD3 positivity in these samples. You see that in some cells, you would observe the cells which are red positive, you would see green staining as well, as indicated by the red arrows. And you also see some infiltration by just CD3 positive T cells. So while the infiltration of the CAR positive cells was studied in the study, what was also evaluated was the antigen. And PSCA antigen expression was assessed in both baseline and on-treatment samples. Since there is no good antibody for PSCA, the ability to see the correlation of PSCA antigen with respect to CAR infiltration in a quantitative manner was the biggest plus point of RNAscope assays here. Very quickly, going into another example of how CAR-T infiltration was studied by Adaptimmune. So Adaptimmune conducted a TCR T-cell therapy in a patient clinical trial, which was later on transferred to another biopharma. NY-ESO-1 immunogenic tumor antigen is expressed in many poor prognosis multiple myelomas. Adaptimmune developed a gene-modified autologous tumor-reactive TCR against the NY-ESO-1 peptide. The use of RNAscope here was to understand the mechanisms of response and resistance to TCR T cells, and also look for functionality of TCR cells post-infusion. What was demonstrated was TCR trafficking to tumor microenvironment, as can be seen here in this image, with the red staining depicting the TCR T cells and the green staining showing the TCR T cells as well as some of the infiltrating immune cells. ISH was performed on FFPE tumor biopsies prior to and following adoptive T cell transfer in synovial sarcoma samples. And these correlated with the anti-tumor effects that they reported. They also showed TCR T cell infiltration into the tumors, as was just mentioned. The ability to see concurrent TCR T cell infiltration as well as immune infiltration was really important for the study. Since these tumors with poor T lymphocyte are known to have poor T lymphocyte infiltration and resistance to checkpoint blockage. And these approaches could help enhance penetrance into tumors to overcome this mechanism of resistance. Lastly, I wanted to share with you one of the examples for CRISPR-mediated gene deletion using our BaseScope Duplex platform. So the aim here was to detect cell-specific Cas9 guide RNA in tissues, as well as to detect CRISPR-edited transcripts in the same tissues. CRISPR-Cas9 was used to delete 49 nucleotides from gene X, resulting in novel junction sequence called as edited. So the top here that you see in the right-hand side panel is the wild type sequence, which included the 49 base pairs of the nucleotides, and the bottom sequence here, which was edited and did not have these 49 base pairs. BaseScope paired oligos, as can be seen here by 2 double Zs, were used and designed to target either the 49 nucleotides in the wild type sequence or the edited novel junction sequence. These images depict how BaseScope Duplex assay is used to study single-cell gene editing expression profiles in the edited and non-edited livers. The top left image shows vehicle-treated mouse liver tissue, which was stained with the wild-type probe in green and the edited probe in red. As you can note, there is only presence of wild-type sequence in the hepatocytes. In the top left image, in the top right image from a CRISPR-Cas9 treated liver, please note the presence of wild-type only hepatocytes as depicted by black arrowheads. The black arrows show the edited livers co-expressing hepatocytes with both red and green dots. Lastly, we showed that the editing was performed in a cell-specific manner, as in the vehicle-treated liver in the bottom left corner image shows no wild-type or edited gene staining in the Kupffer cells represented by the white arrow. And the same goes for the edited liver on the bottom right corner with Kupffer cells showing no staining for the edited or the wild-type gene. This experiment helped establish the cell-specific nature of the gene editing and also helped discern the monoallelic and biallelic gene editing status of cells in CRISPR-Cas9 treated liver. wanted to show you a bird's eye view of these same tissue samples and how the different regions in liver were examined. So the assessment of portal and central veins showed no detectable signal for either probe in endothelial cells, nor was signal seen for either probe in the cells lining the bile duct, or the central vein, or the portal vein. Taken together, these results reveal the CRISPR-Cas9-mediated deletion was restricted just to hepatocytes. This methodology can be applied to various different gene editing and detection studies and help visualize the biodistribution of edited versus non-edited cells. As we close in to the top of the hour, I would just like to share with you some of the accolades that we have received from our collaborators and just what we aim for. A great customer experience from the Pharma Assay Services group here in Newark, California. To conclude, we provide scientific pre-sales expertise to help plan your study with one-on-one teleconferences to understand your goals. Our scientists are available to present the summary data via WebEx to key stakeholders at your site, and a project manager who will ensure timely delivery of your study and that project objectives are met. As we close in to the top of the hour, I would like to share with you some of the frequently asked questions. During this time, please feel free to direct your questions to all panelists in the Q&A window. We will continue to be online for a few more minutes. Feel free to send in your questions to our panelists who are waiting to answer your queries via the Q&A window. As our panelists answer some of your questions, I would also like to take the time to share with you some of the frequently asked questions. You can also visit our website with the link at the bottom for any additional questions. As the questions pour in, would just like to share with you one of the most frequently asked questions was the tissue types that RNAscope technology can be used for. While FFP is the most prevalent tissue that we work with, we are absolutely compatible with fresh frozen tissues, fixed frozen tissues, cultured cells, and PBMCs. Also, there were a few questions with respect to our timelines. What we have shared with you is just a timeline where 20 tissue samples can be stained with 2 or 3 markers. For a project this size, our usual timelines are about 3.5 to 4 weeks. The first part, or phase 1 for sample QC and optimization, takes us about 1 to 1.5 weeks. And during this time, we perform pre-treatment optimization, checking for the ER2 and protease and how well those work for the best signal-to-noise ratio establishment. This is followed by marker evaluation, which takes roughly about a week. And following that, we perform scoring and reporting, which is about a week to week and a half timeline. Of course, if there is halo studies involved, those usually slightly more time and labor intensive and take longer. Our average timeline for a halo-based study, performing just the halo, is about 4 weeks, but of course it depends on the bandwidth of the study as well. Some of the other questions have alluded to the double C probe pairs. As you can see, it does not mean plus or minus strand. It only means how many pairs of oligo probes are in our final probe. Our standard RNAscope probe is about 20 ZZ probe pair, and a BaseScope probe can be anywhere between 1 ZZ to 3 ZZ, depending on the application that we are using it for. Our probe design team can provide more information regarding the strand information for your particular probes of interest. With this, we come to a close to today's webinar. If your questions were not answered during this time, and we will continue to be online for a couple of more minutes, but if your questions are not answered, we will get back to you soon via an email. We will also post a recording of the webinar to our website for any future review. Thank you again, everyone, for joining us. And hope you have a good day and stay safe during this time. For any future inquiries, please direct your emails to the email addresses here, where our teams can help you design your studies. Thank you and bye-bye.